In this video, we are going to talk about amino acids. Amino acids are the monomers, or we can say building blocks of the proteins. All the proteins are made up of amino acids. So many amino acids will get joined to form a polypeptide chain, and that polypeptide chain will be get folded to form a protein molecule, a functional protein molecules. There are up to hundreds of amino acids are present inside the living cells. Out of them, 22 amino acids take part in the protein synthesis, and the proteins are made from those 22 amino acids. In some books 20 amino acids are written, but there are 2 more amino acids take parts in the protein synthesis, or we can say those amino acids are ribosomically added into that protein structure. Those amino acids, 22 amino acids, are called standard or proteinogenic amino acid, and those amino acids are found in the living cells, but does not ribosomically added to the protein chain or polypeptide chain, but they remain in the living cells. Some living cells are known as non-standard amino acid. Okay, non-standard, standard amino acid and non-standard amino acids. So that is what we are going to study in this video in detail, is that those amino acid, the structure of the amino acid, the typical and the general structure of the amino acid, and some how they are get joined to form a polypeptide chain. That is what we are going to study in this series of video. There will be 3 to 4 videos I will upload on this topic of amino acids, and this is related to your CSIR net preparation. So let's start with the basics of amino acids first. So as the name suggests, amino acids, which means there will be amino group present, and the acidic group will also present. So that's why, because of the amino group and the acidic group, the name suggests the amino acids, and the molecule is known as amphoteric molecule, because it can act as a base as well as it can act as an acid. So that's why these molecules are called as amphoteric molecules. So amino acid, if you look at the structure of the typical amino acid, it will something look like this. There will be the carbon, on which on the one side there will be amino group or amino group, whatever you may call it as, amino group or amino group. There will be one carboxyl group. There will be one hydrogen and one functional group. Okay, so these 4 components, these 3 components are common in every amino acid, except some amino acid like proline. There will be NH2 instead of NH3, but this amino group, this carboxyl group and this hydrogen atom is common in every amino acid. Okay, so this skeleton is common in every amino acid. What differs is that this R group, this functional group, is differ in amino acid to amino acid. As the amino acid changes, this functional group will also change. Okay, so this is the general and typical structure of amino acid. You can see here there is the carboxyl group, carboxylate group, and this is the amino group. So that's why it can donate a proton like acid does. So it can act as an acid, and it can accept the proton as well. So it can act as a basis also, because there are both the groups are present. So that's why these molecules are called as amphoteric molecules, right? So this is the generalized structure of the amino acids, and every amino acids has this skeleton common, but this R group is different in amino acid. as the amino acid glycine, in there will be in lysine there will be different R group, in tryptophan there will be different R group, in leucine there will be different R group. So this functional group is different from amino acid to amino acid. So this is the structure of amino acids, right? So we have seen that the amino acids can be of standard. So those amino acids are added ribosomically inside the protein chain, are known as standard amino acid. Those amino acids are present inside the living cells but do not added ribosomically into the protein chain, are known as non-standard amino acids, right? After that, the second point I want to mention in this video is that, let me see, we have seen that okay, so this is the structure of the amino acid and how these amino acids are joined to each other. So one because we know that to make a whole polypeptide chain, to join two successive amino acid, we have to join these two. Suppose we have this amino acid 1 and in this case we have the amino acid 2 and H3, right? So suppose we write this carboxyl group here and this hydrogen is here, right? So we have this hydrogen, this carboxyl group here, this hydrogen here and R group here. So this is the amino acid second. So the amino group of the second amino acid and the carboxyl group of the first amino acid will be react to each other and they will be get joined to form peptide bond between them. A peptide bond will be formed in those two successive amino acid. The reaction will occur in the carboxyl group and the amino group of the successive amino acids, and they will get joined, they will react and they will get joined to form a peptide bond. The bond is known as peptide bond. I am not going into the details of the chemistry, but the peptide bond is formed between two successive amino acid and they get joined. That's why the chain of the protein, the chain ultimately we get after the protein synthesis, after the translation, is known as polypeptide chain. Why polypeptide chain? Because so many peptide bonds are there, so many amino acids are there joined to form a polypeptide chain. So this was all about the structure of the general and typical amino acid and the basics. I have covered some basics of amino acid in this video. For more details, I will make another videos, lecture series on the same topic, amino acids, for you. In those lecture I will cover like optical properties of amino acids, some structures of amino acids, like various structures we can see in the amino acids. So that I will cover in next few videos. I hope you like this video. If you like this video, please hit the like button, subscribe to our channel and share this video to your friends. Thank you so much.